Good day from ChemHelp ASAP. This video describes the discovery of MAK683, which is an inhibitor of PRC2. MAK683 is in development for oncology. This video reports the discovery program of MAK683 as described in a Drug Annotations article in the Journal of Medicinal Chemistry by scientists at Novartis, primarily the research efforts from a team working in Shanghai. The citation is at the bottom of the screen, and a link to the article can be found in the video description. This video is not an exhaustive discussion of the article, but I have selected what I consider to be the highlights that best summarize the research project. PRC2 stands for Polycomb Repressive Complex 2, which is a complex of multiple proteins that plays an important role in chromatin organization. PRC2 catalyzes the methylation of lysine 27, an amino acid residue in histone H3. One of the proteins in PRC2 is called embryonic ectoderm development, EED, and EED activates PRC2 for the methylation reaction. So PRC2 inhibition can be achieved through binding EED, which is a protein component in PRC2. Uncontrolled activity of PRC2 is associated with certain cancers, so PRC2 inhibition should lead to a therapeutic benefit. That is the plan. This particular Drug Annotations article does not begin in a typical manner. Drug programs, especially programs with a known target, often start with molecular libraries and a screening campaign. The details of the early stages of the program were disclosed in a previous Chem article. In that work, the Novartis scientists performed a high-throughput screening campaign with a biochemical inhibition assay. A promising hit was identified through the screen, and binding of the hit to PRC2 was confirmed through an X-ray co-crystal. The hit was subsequently optimized to afford EED226. EED226 was a very promising compound with a number of attractive features. It was selective for PRC2. It had adequate enough bioavailability to demonstrate favorable in vivo efficacy in mouse xenograft tumor models. Unfortunately, EED226 also had limited membrane permeability and solubility. The furan ring also formed reactive metabolites in vivo. Neither article fully details the path of EED226, so I'll do my best to speculate. Based on the information, I suspect the compound was promising enough in animal models to advance into the clinic, but ultimately the permeability and solubility issues prevented adequate efficacious exposure in humans. It's also possible that the metabolism concerns around the furan were not serious enough until the molecule was studied in man. Regardless, at some point, active research on EED226 was halted, and EED226 was relegated to tool compound status. Calling a molecule a tool compound is a nice way to say that the molecule is not good enough to be a drug. The team sought to modify EED226 to optimize further the properties and address the concerns in the lower right of the slide. At this stage is where the Drug Annotations article begins. Here is a slightly different look at EED226 with different parts labeled based on their role in binding PRC2. On top is the surface moiety. This part of the molecule only weakly interacts with the target and mostly projects out of the binding pocket. Being mostly solvent exposed, changes here should not have as much impact on the potency, efficacy, and selectivity of the compound. Next is the core. The core binds within the aromatic cage, a feature within the EED protein of PRC2. In general, electron-deficient aromatic rings provide better binding in the aromatic cage. Finally, we have the furan ring, which sits deep in the binding pocket. In general, electron-rich rings are favored for this part of the compound for interaction with a protonated arginine, R MAK68367 within the backbone of the target protein. Remember these two target features, the aromatic cage and the arginine residue. We'll come back to them in a later slide. For now, the surface moiety of EED226, because it has less critical target interactions, was identified as a possibility for improving the membrane permeability and solubility of new analogs. So expect some changes around this sulfonyl group of EED226. The deep part needed to be explored to avoid the metabolic issues of the furan ring. For much of this video, we will see optimizations of these parts of EED226. Let's start with changes to the deep part moiety. I will note that all these structures also lack the sulfonyl group on the surface moiety. The sulfonyl group has been replaced with a dimethyl amino group. The researchers used this amine as a temporary placeholder because it provided an immediate solution to any solubility concerns. On the screen are 4 different R groups, one of which is the original furan on EED226. The other R groups are alternative electron-rich rings. The different rings all showed some loss of potency in the biochemical inhibition assay of PRC2 relative to the original furan, but the bottom compound fared better in both a cellular assay and a proliferation assay, which measured the concentration at which the compound inhibited cell growth. The Novartis team was able to get a co-crystal of this bottom compound with EED. From this co-crystal, we will be able to see some features of the EED binding pocket. To the left is the full structure of the bottom compound from the previous slide. The co-crystal is entry 7QJU in the protein data bank. The middle image is a space-filling model of the protein bound to the compound, and the right image just shows the binding interactions. Let's take a closer look at each of these representations. Here is our space-filling model. I say space-filling because this representation, these spheres, are of the protein of EED, and bound within the structure is this gray structure. This is our compound that we discussed on the previous slide. So we can see here's our dimethyl amino group, and then the benzene ring that it's connected to, and that is the surface part moiety. As we get deeper into the binding pocket, it's maybe kind of hard to see, but these blue atoms, those are the nitrogens in the core of the molecule. And then as we look even deeper, it's hard to see, but it's down deep in this binding pocket. That is the deep part. That is the electron-rich aromatic ring that is bound deep in the pocket. So the point of looking at this space-filling model is so that you can see that, yeah, there is definitely a pocket in which these compounds are binding on EED. Let's now move forward and look at some of the specific types of interactions between our compound, our ligand, and this larger protein. Here is a different representation of the binding. So now we've stripped away most of the parts of the protein that are not directly engaged with our molecule. Our molecule is shown in a slightly darker gray wire frame, and then the surrounding interacting amino acids are mostly in white. All these lines are interactions between other parts of the molecule. So here is our dimethyl amino group up top connected to the rest of the surface moiety. There are some interactions. You can see there's this line that is a type of interaction between the surface moiety and parts of the backbone, but not a lot of interactions. Once we get down to the core, that's this ring system right here. I want to highlight 2 things. One, there's a ring right here. That is a phenolic ring. That is from a tyrosine residue. And we can see over here, here's another phenolic ring. That's a tyrosine residue. This is the aromatic cage, and you can see that these tyrosine residues are interacting very strongly with the core part of the molecule. And so these electron-rich tyrosines interact with an electron-deficient core aromatic system. Finally, down deep in the bottom, here is our electron-rich aromatic system. This is the deep part, and the deep part is interacting with this residue right here. This is part of an arginine residue. This is protonated, and it sits flat and snuggles in right next to this electron-rich aromatic ring. So that's a key interaction. So we can see all the key parts of interaction between our molecule with its surface moiety, the core, and the deep part, and how it interacts with features within the binding pocket. In addition to trying other rings in place of the furan, the researchers also incorporated a fluoro group on the ring. Personally, when I see a fluoro group in a drug or lead, I tend to think the fluoro has been added to block metabolism, particularly on an electron-rich aromatic ring like this one. The authors, however, observed that introducing an ORTHO-FLUORO group generally reduced activity on alpha-2 adrenergic receptors as well as phosphodiesterase 4D, PDE4D. These are common sources of off-target toxicity, so fluoro substitution was a means of improving target selectivity and safety. Okay, moving to the core of the molecule, the first core scaffold was the triazolopyrimidine system. This is the same as we saw in EED226. Among the different scaffolds explored, the triazolopyrimidine scaffold tended to have more risk of PDE4D activity. Next was the triazolopyridine scaffold. The only change is removal of one of the nitrogens from the ring. The triazolopyridine analogs tended to have higher activity on adrenergic receptors. Finally, some C6-cyano analogs were prepared. The same substitution pattern was seen in the original screening hit that led to EED226. An issue with the cyano series is that changes to the furan gave a dramatic drop in potency. Therefore, compounds moving forward were restricted to the triazolopyrimidine and triazolopyridine scaffolds. Finally, let's look at optimization to the surface part of the molecule. Researchers tried different pyridines in place of the aryl sulfonyl group. While a pyridyl group maintained potency and improved solubility, the pyridine ring was associated with inhibition of cytochrome P450 2D6 and greatly raised the risk of drug-drug interactions. The addition of one or more methyl groups on the pyridine had two beneficial effects. First, additional methyl groups further increased solubility. Second, methyl groups placed adjacent to the ring nitrogen greatly reduced CYP inhibition. So we've now seen the collective main changes considered for the tool compound EED226. Let's now see some of the most promising leads. From the lead optimization campaign emerged compounds 21 and 22. Note that 21 has the triazolopyridine core and 22 has the triazolopyrimidine core. Each compound also has slight differences on the other parts. Based on in vitro assays, 22 shows better potency in the cell proliferation assay and better solubility in simulated gastric fluid. Compound 21 demonstrates somewhat better membrane permeability in the CACO2 assay. In vivo PK studies in the rat showed that 22 had lower clearance. Volume of distribution was comparable, so 22 had a longer half-life than 21. 22 also had better oral exposure based on both AUC and Cmax. Here are some in vivo efficacy data, both in the mouse and the rat. I only mention results from 22 on this slide. Compound 21 was also tested in mice, but the data for 22 were better, so only 22 was advanced into rats. In a mouse xenograft study, 22 was dosed twice per day orally at 4 different levels. All 4 dosing groups showed complete remission. While all 4 groups had the same outcome, higher doses did result in faster reduction in tumor size. Compound 22 was well tolerated, and all animals remained tumor-free through 68 days, the end of the study. These animals were considered to be cured. Because of the promising results from the mouse study, the dosing levels of 22 in rats were reduced. All groups except for the lowest, 0.3 mg per kg administered orally 4 times per day, showed complete remission. Again, 22 was well tolerated, and the rats remained tumor-free for over 130 days. Based on the in vivo efficacy data, compound 22, research designation MAK683, was studied in preparation for clinical trials. Preclinical safety studies, including hERG channel activity, mutagenicity, and cytochrome P450 inhibition, were all favorable. MAK683 had reasonable predicted human clearance and volume of distribution. These values would give an observed half-life between 3 and 4 hours. The predicted bioavailability was 73%. The predicted efficacious daily dose was 88 milligrams for a 70 kilogram patient. At the time of this video, MAK683 is in an active Phase 1, Phase 2 trial. The clinicaltrials.gov identifier is on the screen. The study specifically lists diffuse large B-cell lymphoma and nasopharyngeal carcinoma as potential indications. Thank you so much for watching to the end of this drug discovery story on MAK683. Please consider subscribing to the channel, leaving a like, or making a comment. Take care.